Well, welcome everyone to our first virtual meeting in 100 years. We were expecting our 100th anniversary year, and this is the first time we've been able to meet. Just a few months ago, Tom Moses' widow was going through his papers, and she found the original Technical Club of Madison bank account book. And the first entry was in spring of 1920. So even though the club wasn't incorporated until 1921, it seems like they were doing things in 1920. So I'm glad we were able to at least have our first meeting in our 100th year. And usually I would welcome the guests here. If there, I see some guests in the background, welcome to our meeting. And I'm very grateful to Professor Dubielzig. And I'm going to turn it over to him. And let me just talk about the questions. So I think we're going to ask everybody to mute once Dick starts his talk, but you can send the chat. It's set up so they'll all go just to me, and then I can write any questions down, and then at the end of the presentation, I'll read those questions back, and then maybe we'll have time for more questions you might have as we go along there. So I will now turn the meeting over to our speaker, Professor Dick Dubielzig, who's emeritus professor in the veterinary school. And I will put the spotlight on Dick.
Okay, I'm going to go to screen share.
Oh yeah, hold on, I'll open up the screen. Oops. Yeah. Whoops. Did that work?
There you go.
Yeah.
Is that good for everyone? Can everyone see the screen share?
Looks good.
They're all muted.
Yeah. So my name is Dick Dubielzig, and I am a professor emeritus of pathology at the veterinary school, as was described. Madison, Wisconsin is my hometown, and I came back to be a part of the Department of Comparative Biosciences in the veterinary school in 1983, which is the year the school started. Actually, I was the very first faculty member in my department to be on board. When I came, I had been at the University of Pennsylvania, and I already had a particular interest in ocular pathology. But the job I was hired to do was to be a general pathologist, to participate in the necropsy and biopsy service at the veterinary school. One of the conditions that I made when I was hired was that I would be allowed to have a fee-for-service account and receive ocular tissues for anatomic review, and then I would have control over that fund. So I started in 1983. It wasn't until about 2003 or 2001 when my children left the house to go to college, and I realized that my wife was a 8th grade science teacher, and she's also a perfectionist, which means that she was working all the time. So with the kids gone, I realized that I had an opportunity. I had more time than I ever had before. I had an opportunity to make ocular pathology more than just a part-time hobby. So for the last 20 years of my career, ocular pathology gradually, first one step and then the other, gradually became not just a hobby in my work, but became the central part of work. So the lab that I started with a fee-for-service foundation is called the Comparative Ocular Pathology Lab, COPLAO. and you can see the logo in the lower right. So we receive specimens of ocular tissues. About 2/3 of them are actual globes, actual eyeballs, and the other third are eyelids and tumors or other diseases around but not directly in the eye. And all that time, in addition to having an interest in the pathology of diseases of the eye, I developed more and more an interest in comparative anatomy. So we always encouraged zoos and aquariums and wildlife biologists and scientists and other people interested in comparative anatomy to send us specimens of a wide variety of different types of animals. And there's no way of knowing this for sure, but I wouldn't be surprised if we have the largest collection of ocular anatomy, number of different species of ocular anatomy anywhere in the world. It's although that remains an interest of mine, it's never really been the core of what we do. So my manuscript portfolio in comparative anatomy is much less than my hobbyist interest in the topic. So I'll start out with a disclaimer. I am not an expert in optics. The knowledge that I bring to comparative ocular anatomy is strictly that of an anatomist. There are probably people here who understand optics more than I do. I also don't understand the cell biology of the tissues of the eye as well as I might. But what I do know is what we get to see in the lab, and that's the comparative anatomy. So this talk is going to be, I'm going to go over the variations in anatomy that we see in the eye dependent on the use or the function of the eye in any particular animal. So I'm going to start out with adaptations in ocular anatomy that are useful in optimizing visual acuity. And if in animals that have outstanding visual acuity are almost by essence going to be animals that use their eyes during the day. It's a contradiction in terms to have good vision in dim light and also to have maximum amount of visual acuity. So I've put on the screen here the eyes from two completely different groups of animals, both of which are exquisitely adapted for maximizing visual acuity. On the right is a red-tailed hawk. If you're looking for adaptations for visual acuity, first of all, birds are the group of animals you want to look at. And I gave a talk on the incredible vision of birds. And then second, among the birds, the diurnal raptors, the birds of prey that hunt during the daytime, are the ones that are best adapted for visual acuity. So I'm not going to talk too much about visual acuity in birds. I'll just mention it here and there. On the left is a mammal that has eyes that are adapted for a high degree of visual acuity and also for a diurnal lifestyle, and that's the cynomolgus macaque as an example of a primate eye. Basically, most primates, not all primates, but most primates among the mammals have perhaps the highest degree of anatomic adaptation and functional use as instruments with high visual acuity. If you want to adapt an eye to have excellent visual acuity, some of the features that you need to have are a big eye, first of all. Now, the disclaimer there is a big eye is also useful when you want to have vision in a dim light circumstance. But the bigger the eye, the better the eye it can act as an instrument for visual acuity. One of the reasons for that is what you want to do is you want to have cast on the retina is in back of the eye. Here it's this tissue that picks up the focused image. You want to cast on the retina as large an image as possible. So in order to do that, what you want to have is you want to have the optical center of the eye. The tissues of the eye which focus light are the cornea here and the lens, both acting together. So you want to have this optical center as far forward as possible. And what that means is that the image on the retina can be as large as possible. It will be diffused, being large, but remember that animals that have high degree of visual acuity function during the daytime, and the amount of light available is not a problem. So animals that see well in the daytime, generally speaking, tend to have a small cornea, and by that I mean the percentage of the circumference of the globe devoted to cornea is relatively small. They tend to have a forward positioned and relatively flat lens, and then they tend to have some part of the retina that's specialized for a high degree of visual acuity, and that structure is referred to as the fovea. If you take a primate eye and draw a line right through the center of the eye, this is the area where you'll find the fovea. It's not very well illustrated in this specimen. The diurnal raptors, just as an aside here, actually have two fovea in each eye. It's not very well known how they use those two foveas, but that is something that gives them excellent visual acuity. So this, I've gone from a macaque to an orangutan here. So that's basically the same organized principle of the eye. Now you can see the fovea a little bit better. In some specimens, you can see a little yellow area around there. It's fovea, the macula. The macula is this expanded area that's used for a high degree of visual acuity. The fovea is this actual little pit in the microscopic images on your right. You can see the sections right through the fovea. But the yellow coloration is an adaptation to protect against ultraviolet light. And although this specimen is fixed in such a way you can't see the color of the lens, animals that see in bright light often have yellow pigment in their lens, again, as a mechanism to protect the back of the eye against ultraviolet light. So these are three examples of what you see under the microscope in the area of the fovea or the macula. And you can see it consists of a pit. When you're looking at let's use this bottom image here. When you're using the retina to analyze light, the light interacts with the tissue in the far back of the retina. So in other words, light has to pass all the way through all the layers of the retina, and then it interacts with this layers back here known as the photoreceptors. And then the photoreceptor cell not only is these layers here, these two layers here, but also this layer of nucleus here, which is the outer nuclear layer. And then there's a series of synapses, and the cell that actually transmits information to the brain, we can't see in this image because it's so focused on the fovea. But if we look at this upper image, you can see this blue layer right there. And those are the ganglion cells. And what I want to highlight here is how many ganglion cells there are in the primate retina in the area adjacent to the fovea. As a marker of visual acuity, the number of ganglion cells is a good indicator of a portion of the retina that's used for high degree of visual acuity. So this is a couple of other ways to look at the eye. This is what's called a multifocal electroretinogram. So a light is flashed in the eye with a pattern, and using that pattern, a computer can calculate what the electrical activity is that comes from multiple little individual portions of the retina. So this is actually human. what we're looking at here is the amplitude of the electrical activity. And in the area of the fovea, the amplitude of the electrical activity is greatly enhanced. You wouldn't see that in an animal that's adapted for diurnal light, although you definitely would see it in a bird. This doesn't tell you exactly how that electrical activity is being used or processed. It's just a rather coarse illustration of the relative function of the retina in different parts of the retina. This is a topographical map, and what has happened here is the retina has been removed from the animal. Let's say this is a monkey. Retina has been removed from the animal. It's been cut so that it can be laid flat on a slide, and then it's been stained so that you can look through it with a microscope and actually count the number of ganglion cells. Remember, the number of ganglion cells, the cells which provide information to the brain, is an estimate for the part of for the visual acuity, or the function of that part of the retina that's good for visual acuity. And so now you can see this topographical map, number of ganglion cells, that there's a concentration of ganglion cells in the area of the fovea, which we had already seen. I'm not going to get into this, but this is a relatively new technique for evaluating the retina in vivo, in a live animal. It's used extensively in medical ophthalmology to evaluate the morphology of the retina. Highly useful structure. So I think I showed this image the last time I talked, and it's a gradient of visual acuity calculated by a number of different techniques. And it shows that here you have your diurnal raptors on one side and your nocturnal birds on the other. And it shows that the human vision in terms of visual acuity is actually right up there among the birds. It's not among the top birds, but it's right up there among among the birds. There's nothing special about the poor visual acuity of a cat. It's fairly typical of mammals. Mammals in general, primates are something of an exception, but mammals in general have eyes that are adapted for nocturnal or dim light, functioning well in dim light, but not necessarily forming the highest degree of visual acuity. And the reason for that is probably fairly well documented. The reason for that is that during the dinosaur era, mammals were forced to find niches where they could avoid dinosaur predations. And they did that by either going underground or hiding in caves or coming out at night or populating trees. And in the process of doing that, they actually lost visual function in the eye. Now, how do we know that? And the reason is because there is a mammal that has anatomical features that date from way back in mammalian evolution. And those mammals, although they have relatively poor vision, have some of the morphologic features that are associated with high degree of vision. And those are the monotremes, in particular the duckbill platypus. If I don't ramble on too long, I have some pictures of duckbill platypus eyes in the back of the talk. Okay, so not so much for pure visual acuity.
I wonder if I could ask a question.
Absolutely.
You said the mammals, and as a group, lost visual acuity because of selective pressure. Did they gain nighttime vision improvements at the expense of visual acuity?
Absolutely. Mammals, in comparison to birds, which are mostly diurnal, have eyes that are adapted morphologically for nighttime vision. Now, having said that, both birds and reptiles have eyes that have some characteristics of sophistication that even though they're perhaps moderate in terms of the differentiation purely for dim light vision, also are able to maintain good dim light vision. Owls, for instance, and geckos have eyes that are excellently adapted for dim light vision. Okay, another feature somewhat related to visual acuity is the ability to react to a visual stimulus extremely rapidly. In other words, to have very fast reactions. And when you're talking about the eye, the way you enhance the ability to react to motion very rapidly is to enhance what's known as the cone flicker response. Now, the cone flicker response is a measure of how quickly the cone is the photoreceptor that functions in bright light, how quickly after being exposed to light, the cone is able to replenish its photoreceptor and then function again. We know, for instance, in a movie camera, I forget what it is, it's however many frames per second, a human eye is no longer able to recognize individual frames, and the human brain then interprets the motion that occurs as being not interrupted. Most birds have a much more rapid cone flicker response, and even a lot of mammals have a much more rapid cone flicker response than the human eye. Human eye is not particularly good at having a very rapid cone flicker response. Among the birds, there are some that depend upon extremely fast reflexes, and we were working with a group who does that topographical mapping of ganglion cells, and they discovered something unique and interesting in the flycatcher retina. And just for reference, I'm gonna try to play a YouTube video and see if it will come up on this Zoom talk. So bear with me. So we're looking at here. Oh, let's do that again. What we're looking at here is a flycatcher sitting on a branch, which is how they catch their insects. They just sit there, wait for an insect to fly by, and then catch it in their tiny little beak, which I think is astounding. So this is one of those topographical, one of those flat mounts of the retina of a flycatcher. And what we're looking at is not the ganglion cell layer, but the photoreceptor layer. And lo and behold, there's nothing special about this light that has caused the coloration. The color that you see here is natural. It may have been enhanced, but it's natural. And you can see there are these bright red conical structures, and then there are also varying colored round structures. Some of them clear, some of them kind of yellowish green, some of them kind of blue. And these structures are known as oil droplets. And oil droplets are a feature of many different kinds of mammals. Many different kinds of non-mammalian vertebrates. And then let's throw in the duck-billed platypus in that too. This is one of those examples of a ocular sophistication that's maintained in that primitive mammal that you don't find in other mammals. So there's nothing special about that. I'll talk a little bit more about those oil droplets in a minute, but let's concentrate on these conical structures. If you look at them up close, they almost look like a dome light with some sort of mechanism in the center. And so what we did, our participation in this is to look at the electron microscopy, where you can look at a much higher detail. And this is what we saw. We saw in the outer portion of the photoreceptor, we saw this structure lined by these little vacuoles that can almost certainly contain the red oily material that gives it the red color. And then in the center, there's a gigantic mitochondria. And the mitochondria is a structure which provides the energy for any cellular feature. And like I said, this cone flicker response is a mechanism that if you're going to enhance it, it requires an incredible amount of energy. The photoreceptor cells in general, cone flicker regardless, are the most energy dependent, the most oxygen dependent cells in the body. One might guess, if this hasn't been measured, but one might guess that these specialized cones, which have the ability to see and refurbish very fast, have an additional energy requirement, and hence these giant mitochondria. Now, giant mitochondria in other cellular tissues have been shown to be responsible for enhanced energy production. So if you have an extremely rapid cone flicker turnover, then what you're able to do is like a slow motion camera. You're basically able to your brain then is basically able to calculate whatever is happening in relative slow motion, giving the flycatcher an added advantage. This is now not electron microscopy, but just regular microscopy, and these dark structures here are the equivalent of what we saw. Notice the oil droplets, these little clear areas here in the flycatcher as well. You don't need to see that. You don't need to see that. Okay, so slow motion vision. So let's, I said that birds in general have a high degree of visual acuity. Let's go back and talk a little bit about reptiles. Now, dinosaurs, which dominated the era, the Jurassic and the Cretaceous eras, are derived from reptiles, and then birds are derived from theropod dinosaurs, so that you can think of them in sort of the same way. But reptiles, as they were evolving, are cold-blooded animals, and the issue there is that in order to warm up their body, they need to be out in the bright daylight. And if you're going to evolve eyes that function when you need to be out, you're probably going to evolve eyes that have visual function under bright light circumstances, and then that's just as if it's just natural then that probably the natural adaptation or competition would suggest that you develop more and more high degrees of visual acuity, although they can be specialized in other ways as well. So many of the features that we see in bird eyes also occur in reptilian eyes, and reptiles in general have eyes that are adapted for a high degree of visual acuity and a high degree of function in bright light circumstances. This is a turtle retina, and this is the same thing. This is a flat mount looked at through a microscope, focusing on the photoreceptor area, and the light and the color that you see here, again, is not artificial color. It's enhanced color, but it's not artificial color. And you can see that there are 1, 2, 3 for sure. Maybe this is a fourth color here in this turtle retina. And so the turtles and birds have at least 3, and in many cases 4 different types of color vision specific cones, whereas mammals as a rule have 2. The primates, which I've already said are adapted for a high degree of visual acuity, and that is to say the old world primates, the primates that evolved in Africa, are the only ones that have 3, like for instance us. So, but we don't have any oil droplets. The advantage of having an oil droplet is that the color of the filter is exactly the same as it filters out all the light except the light that's exactly needed for that particular type of photoreceptor, which sort of fine tunes the ability of the photoreceptor to differentiate between different wavelengths of light. So that is a feature of both birds and most reptiles, even goes back into amphibians and fish. And like I said, among the mammals, I'm only familiar with the duckbill platypus. There's another monotreme, the echidna, which is derived from the duckbill platypus, and I expect that it has oil droplets as well, but I don't have an example of an echidna eye. So reptiles, as a general rule, have a fovea. Reptiles, as a general rule, are diurnal animals, and so they use their fovea in the same way that primates and birds use it, as a portion of the retina that's developed for a high degree of visual acuity. The tuatara, by the way, is the most primitive of the lizard family of reptiles, and even in that extremely primitive reptile, we have a fovea. One of my favorite eyes of all is the eye of the chameleon. Now, the chameleon has eyes that function in the daytime. They're diurnal eyes. They have a high degree of visual acuity. They catch insects with their tongue, and if you've ever tried to catch an insect with your tongue, you might guess that they require a high degree of depth perception, a very accurate depth perception in order to launch that tongue. And so there are a number of very peculiar features of the chameleon eye. One of them is that they have independent eye movements. Probably most of you have seen videos of chameleons where their eyes go wandering around in different directions all at the same time. They have depth perception that doesn't depend on using both eyes. They have monocular depth perception. And the reason for that is they're able to cast a very large image on their retina, relative to the size of the eyes, a very large image on their retina. And they're able to perceive the depth of a structure simply by adjusting the amount of focus that they have to do in order to get that image in focus. They're the only vertebrate that has a negatively powered lens. What happens if you have a negatively powered lens, it means that all of your focusing has to be done with the cornea, but it means that the optical center of the eye is pushed forward like a telescope, same principle as like a telescope. And they have a very high degree then of corneal accommodation. Accommodation are the mechanisms by which you can see things that are up close. And mammals in general have extremely poor accommodation. Primates have, like us, who do a lot of holding things in their hands and looking at things up close, have probably the best accommodation of any of the mammals. But birds and reptiles far outshine any of the mammals in terms of their ability to see things that are up close. If you think of a woodpecker going up and down a tree and looking for insects about a centimeter or less than a centimeter from where its eyes are, it's not using touch, it's not using smell, it's using vision to find little things in the bark of a tree. And then because most of the chameleon eye is devoted to one big fovea, they also have tunnel vision. They only see a small portion of their visual field at any one time. So let's build on that a little bit. I'm going to show a video of a small portion of a video of a chameleon.
Is back.
We're not seeing the video, Dick.
So I love showing that video to people who study the neurology of the eye, because the control of the binocular control of the eyes is a very precisely monitored thing in the human brain, or virtually all other vertebrates. But the chameleon seems to be able to use each eye independently. So I don't have a picture that shows it very well, but the chameleon lens actually has a divot in the front, which makes it a negatively powered lens. But what I want to show here is that the chameleon retina is basically nothing more than one big fovea. And I think that shows up a little bit better here. Now you can see the yellow coloration, which is protective against UV light. This is the retina. The retina only goes from here to here. and most of it is made up by a gigantic fovea. In this section, you can see the fovea right there. So what that means is that the chameleon has an incredible degree of tunnel vision. It can't take in its entire environment visually. And since the chameleon is both a predator and a prey species, it needs to be able to focus intensely on an insect that it wants to catch with its tongue, but it also has to be able to monitor its entire environment up and down and all around, because it lives in the branches. And so that's why it's adapted this technique of being able to move its eyes independently to survey the environment around it. Actually, when it strikes, it uses both eyes at the same time. I'm told by people who study chameleon neurobiology that even when they focus both eyes on an insect, they're still using each eye independently with monocular depth perception. I don't know how they know that. Look at how thin the cornea is, for seeing things up close, the chameleon's got to be able to change the curvature of the cornea, and it does that with the only muscle that it has in the eye. That's this muscle right here, which is attached to this long tendon, which is attached to the most peripheral portion of the cornea. And since the cornea is so thin, and if you pull on the most peripheral portion of the cornea, the central portion of the cornea, which is the focusing, where the rubber hits the road in terms of focusing, becomes much more powerful as a focusing apparatus.
Is the narrow aperture an accommodation to allow more? An adaptation to allow.
Talk about this.
Oh, sorry. I said, is the small aperture of the chameleon eye an adaptation to increase range of focus?
So yes, the chameleon has a very small cornea compared to its eye size, and is capable of having a very small aperture in its pupil, which of course will give it a much greater depth of field. So I believe the answer to that is yes. It's not unique to the chameleon, though.
Thank you.
Okay, let's talk about a species that has a high cone retina, but it doesn't use that high cone retina particularly to maximize visual acuity. This is a species that wants to enhance its ability to see in bright light. If we were out in on the prairie and looking for a red-tailed hawk, we probably wouldn't look this close to the sun to try and find a red-tailed hawk. But the main predation for the ground squirrels, all manner of different types of ground squirrels, is raptor predation coming from above. And what ground squirrels do is they come out of their burrows. They spend their downtime underground in a burrow. And then whereas most of the squirrels are foraging, one or two squirrels will be the sentinel squirrels, and those squirrels spend their time surveying the environment for danger. And the area that they're most concerned about is above. And hawks, diurnal raptors have learned that it's a good thing to dive for prey with the sun directly behind you, so making them even harder to see. So ground squirrels of all different types have eyes that are adapted to be able to function in a high degree of bright light. So how do they do that? So here's one of these topographical maps. This streak here is actually the optic nerve head. In most mammals, the optic nerve head is flat. And these are some raw images from different types of squirrels. There's a red squirrel, a prairie dog, a 13-line ground squirrel, a western squirrel, which is a tree squirrel. That linear optic nerve head is not so great. This is a groundhog. And then this is a closely related species, the flying squirrel, which happens to be different than all of the other squirrel families in that it's very strictly nocturnal. So it has a round optic nerve head. So here's that in the topographical map. Here's that optic nerve head, and you can see that the highest degree of ganglion cell density is just below the optic nerve head. So the optic nerve head is placed high in the eye. This good example on this ground squirrel or the red squirrel, and that means this portion of the lower retina is used to scan the sky, used to look at the sky. So they have eyes that are adapted at rest to be looking at the sky. This is the the eye of a prairie dog, and what jumps out at you right away is the bright yellow coloration of the lens, which is a protective against ultraviolet light, which might damage the cells of the retina. And here is a microscopic view passing right through that streak where the optic nerve head is, and you can see how narrow that streak is. You can see that there are lots and lots of ganglion cells, as you'd expect in an animal adapted for function in bright light or a high degree of visual acuity. And then what you can also see is that the nuclei in the back of the cells in the back of the lens, which is called the outer nuclear layer, these are the nucleus of the photoreceptors, and they're relatively narrow. And what that means is that most of the photoreceptors are cones, useful in bright light. For a long time, it was thought that the ground squirrel, the 13-line ground squirrel, had a pure cone retina, but that's not even close to being true. They still, even though they have many, many cones, they still have more rods than cones. It's just that the rod outer segments are extremely thin, and you have a hard time seeing them. Now, let me point out another feature here. This is the pigment. This is these cells here are called the retinal pigment epithelium. And in birds and in reptiles and fish, the retinal pigment epithelium actually shades the outer segments of the photoreceptors. That's a feature that's been lost in all of the mammals except the duck-billed platypus. But it's also been reestablished in the ground squirrels. And it's a great advantage because if you can shade the photoreceptors, the portions of the photoreceptors that interact with light, the portions that are most likely to be bleached if there's too much light, then you can enhance your ability to see under dim light circumstances. These are electron micrographs, and again, you can see here's what's called the outer segment. Here, this is where the light interacts with the photoreceptor cell, and you can see the pigmentation, the melanin pigment comes up and surrounds the outer segments of the photoreceptors. Now, this is kind of tricky. This is a ground squirrel. And this electron micrograph was taken from a retina that was harvested from a ground squirrel that was hibernating at the time that it was sacrificed. And because the retina is a tissue that consumes a lot of energy, whether it's being used or not, whether there's light functioning on it or not, the hibernating ground squirrels have developed an adaptation to turn off their retinas, and there's actually a structural manifestation of that. I'm not going to go into the details here, but the outer segments of the photoreceptors are manifestly different than the functioning ones in the summer squirrel. There's another mechanism that's been that's helpful for animals that have to see in bright light, and this is mostly seen in ungulates, in hooved mammals, and that is something we call the granular iridica. It functions like a sun visor to keep the sun out of the eye, and you'll see it in most prominently on the upper iris, immediately adjacent to the pupil. But you can also see it sometimes in the lower iris as well. And probably the animals that have the best adaptation of a granular iridica are the camel family, which includes both the llamas and the camels. But the horse has a granular iridica, most often called the corpora nigra. Many other forms of ungulates have it. Exceptions are the pig family, the pigs and the peccaries. Actually, cetaceans, the whales accurately classifying them in their phylogeny, that would go along with the ungulates. Whales don't have so much a granular reticle, but they have another adaptation, which I'll show you. The only other one that doesn't, it's just the pigs that don't have a granular retica. And if you look at their lifestyle and calculate how much shade they have that they live in, the animals that live with the least shade are most likely to have a large granular retica. Okay, so let's look at some adaptations that are helpful for nocturnal vision in dim lights. And this is, like I said, sort of the specialization of the mammal, although it's not strictly limited to the mammal. So this is a graphic from a wonderful book written in the 1940s by Wall, which is basically a book about comparative anatomy of the eye. And it illustrates in cartoon fashion the retina from an animal that has diurnal vision, it functions in the daytime, and a retina from an animal, a standard retina from an animal that has nocturnal vision, functions at nighttime. And the first thing to notice is that the diurnal animal has mostly cones, which take up more space. Therefore, they have fewer nuclei of the photoreceptors, and that's the outer nuclear layer. Whereas the nocturnal animal has more rods, which are very narrow and also very long. And because each rod outer segment has a nucleus, there's going to be many, many more nuclei. The outer nuclear layer will be thicker. And then let's jump to the ganglion cell layer. Ganglion cells, remember, are the cells that send information to the brain. In the diurnal animal, especially if visual acuity is maximized, you're going to have more ganglion cells because you're going to send more information to be decoded in the brain. Whereas in your nocturnal animals, your quality of vision is not so good. It could well be that that detection of motion is prioritized over detection of form, and you don't need to send as much information to the brain. Ganglion cell numbers decrease rapidly. So those are kind of some kind of a hint. And then let's look at, remember I told you for visual acuity, you want to have a relatively small cornea, a relatively flat forward position lens, and then some sort of function in the retina for visual acuity. The opossum is an animal that's exquisitely evolved for vision at night. They're almost exclusively nighttime animals. Notice that the proportion of the circumference of the eye that's devoted to cornea is much expanded. That means that they can see, they can use light from all different portions of their environment. They don't have any part of their retina that's specifically for high visual acuity. They can become aware of things that are happening, perhaps not with visual acuity, but perhaps with motion detection in a bigger portion of their environment. And then most mammals, the default of most mammals is to have in-between eyes. So they have good vision they maintain good vision in the daytime, but they also maintain good vision at periods of dawn and dusk when there's limited amount of light, but probably not the best vision in the world in the deep darkest of night, or in caves or other in such environments. So here's two eyes from very closely related animals, the leopard cat and the cheetah. The leopard cat is not a leopard, it's very closely related to the fisher cat, and they're both exquisitely diurnal hunters. And then the cheetah is a diurnal hunter. Did I say diurnal? They're exquisitely nocturnal hunters, but the cheetah roams the savanna and hunts using its speed and hunts exquisitely in the daytime. So let's look at their eyes. The leopard cat has a huge, I call it a picture window cornea, to let in as much light as possible. They can also dilate their pupil to let in as much light as possible. The lens is much rounder, and the optical center of the eye is positioned almost in the center of the eye. And then, of course, there's no area centralis. There's no portion of the retina that's adapted for high degree of visual acuity. The cheetah, on the other hand, gets to be almost like the primate. It has a relatively short cornea in terms of percentage of the circumference. It has a relatively flat lens, not as flat as the primates, mind you, but has a flat lens. And then there's not a fovea, but there's what's called a visual streak, which is an area of enhanced numbers of ganglion cells. Okay, let's do the same thing with our old friends, the squirrels. Here is the prairie dog, and here's the retina of the prairie dog. Lots and lots of ganglion cells, relatively few photoreceptor nuclei. And here's our friend, the flying squirrel, a big picture window cornea, a relatively round lens positioned more centrally in the eye, and then a very thick outer nuclear layer and extremely long, mostly rod photoreceptors. Flying squirrels are an exquisitely nocturnal mammal. Okay, I'm not going to talk too much about nocturnal birds. There's a whole lot that's written and known about how owl eyes are adapted for nocturnal vision, whereas most birds have a very rich cone, a very cone-rich retina. The owl does have cones, and it does have reasonable vision in the bright daylight, but most of its photoreception is carried out by rods, so it has a high degree of visual acuity at night. A nice estimate of how important vision is to a species is to make a preparation like this, where you dissect away the skull until you have nothing but eyes and brains and beak left. And this is I think it's a great horned owl dissected in such a way, and you can see that there's the owl devotes a whole lot of very important head space to holding the eyes. The eyes are so closely held in the eye socket that there's no room for the eyes to move independently. And that's one of the reasons why owls are able to move their head around 80 degrees so they can look, basically look backwards. They also have what's called tubular eyes. They have, compared to other birds, they have a massive bone in the center of the eye, which holds the eye in a tubular space, increasing the distance of the eye. So owls are among the very, very few animals that have adaptations for nocturnal vision that also have a fovea. So it's not completely true that the mammalian adaptations for nocturnal vision surpass what can be achieved by other non-mammalian animals. It's just that adaptations such that the mammals have are largely adaptations which make their eyes functional in dim light, so under dim light circumstances. Here's the owl. Here's the fovea from an owl. You can see how long the photoreceptors are. These are mostly rod photoreceptors, and yet there's still large, because it's a fovea, there's still large numbers of ganglion cells, probably giving this owl, even at night, a high amount of visual acuity. The snowy owl is an exception. The snowy owl is a diurnal hunter, and that's just in there as a sort of a factual. Interesting, among the nocturnal birds, your owls and nightjars and frogmouth hawks share another feature, which I'm not completely sure I understand, and that is the muscles of the eye. There's two muscles of the eye. One muscle is responsible for accommodation on the lens, and then the other muscle is responsible for accommodation on the cornea. And in the nocturnal birds, the muscles responsible for accommodation of the lens are greatly reduced. Here, for instance, is what's called Brooks muscle. There's only basically one muscle fiber sampled in this planar section, whereas what's called Crampton's muscle, which accommodates using the cornea, is relatively massive in the nocturnal birds, and that's true in all the different types of nocturnal birds. And I can speculate, but I don't have a great idea as to why that is. We also have nocturnal vision in reptiles, and the gecko is a great example of that. Geckos as a group are extremely ancient. They go back before dinosaurs, still recognizable as geckos. and yet they maintain more or less the same sort of body pattern. Now, most geckos that are extant, most geckos that are alive today are in fact nocturnal. They're nocturnal hunters. There are also diurnal variants, but remember that geckos came from reptiles, and reptiles, because they're cold-blooded, developed vision that was functional in the bright sunlight. So they developed a cone-rich vision, and night vision in the gecko is achieved by changing the function of a very cone-rich retina to photoreceptors that can function in dim light. And one of the factors associated with that is that geckos maintain a high degree of color vision, even when they're functioning in under dim light circumstances. Okay, differentiation for dim light vision. Let's talk about the tapetum lucidum. If you've been in the dark and you've shined a flashlight, or you've taken a flash photograph of your dog or a bunch of other different kinds of mammals at night, you know that there's this phenomenon called eye shine, where the eyes shine brightly back at you. And that's because basically they have a mirror in their eye called the tapetum lucidum. And the tapetum lucidum functions by reflecting light that passes through the retina, causing it to pass a second time through the retina and enhance the probability that an individual photon will be picked up and can be used for some sort of useful vision. Maybe not necessarily form recognition, but motion detection would be another example of vision that would be useful at night. Now, the tapetum mostly occurs in animals of a particular giant clade, phylogenetic group. And within that group, there are basically two different kinds of tapetums. There's a fibrous tapetum, which is what you see in a horse, in your cows and cows and deers and other animals that have a rumen. Remember, the cetaceans, the whales, are also in this ungulate group. They have a fibrous tapetum, but they're not found in pigs or camels. The cellular tapetum, which is a variant of this, are found in your carnivores and all the different types of carnivores. Whereas the fibrous tapetum, the reflective element of the tapetum is collagen. In the cellular tapetum, the reflective element is a combination of protein and zinc, or protein, zinc, and cysteine. And basically there are two different types of cellular tapetum, those that are found in the dogs and most other carnivores, and then those that are found in cats, in which riboflavin is also one of the reflective elements in the tapetum. And I put the word hyena in here to remember to remind you that the hyena is actually phylogenetically is more closely related to the cats than it is to the other animals. The tapetum functions to enhance vision when you're looking at dim light, and in most mammals that have a tapetum, the tapetum is in the upper side of the eye, and that's because when the animal is evaluating a environment with dim light, it's focusing on the ground, because the ground is always going to be dimmer than the sky. Whereas there's no tapetum on the lower portion of the fundus of the retina. And that's the portion that would be devoted to looking at the sky or the upper elements of the environment. So here we have a cat, and the tapetum is this bright area here. We look with a fundus camera, with a camera to see the back of the eye. And here we have a dog where the color happens to be green. And this is just a microscopic view. This is a view of a cellular tapetum in the back of the eye. And with electron microscope, the actual reflective elements are incorporated in the structure of these intracytoplasmic rodlets that incorporate protein and zinc. And then here's just an example of eye shine. I'm not going to talk about this picture. This is one, same thing in the cat. The cat tapetum has a little bit of a brown color, maybe because of the riboflavin. Because there's riboflavin in that tapetum, it also expresses what's known as autofluorescence. If you shine UV light on it, it will give off a green light. And I'm not going to show the other one. The pinniped, also actually the pinniped and the cetacean are an exception to the rule that the tapetum is going to be in the upper portion of the retina. Remember I told you the tapetum is in the upper portion because most animals that live on land have a dim view when they're looking at the ground and a bright view when they're looking in the air. But if you go underwater, there's no longer a horizon to divide dim versus light. And most animals that both seals, the pinnipeds, and the cetaceans, the whales, want to have vision at deep elements. So they want to have vision in dim light under all circumstances. So they have a tapetum that covers the whole back of the eye, both of them. Although one is a cellular tapetum, the seals, and the other one is a fibrous tapetum, the pinnipeds. I think that's all we need to talk about that. We also, on occasion see tapetums in other forms of mammals. We see a riboflavin tapetum in primitive primates, your lemurs and bush babies and such things. They have eye shine at night, and it's made up not of riboflavin that's incorporated with protein, but with crystallized riboflavin. And the hallmark of that is sometimes when you receive an eye in fixative, everything will be discolored yellow because that riboflavin has leached out of the tapetum. And then there are a few rodents, not many, but a few rodents that have a fibrous tapetum. And then the opossum, you've probably been driving along at night and you've seen the eye shine of an opossum on the side of the road. The opossum actually has what's called a retinal tapetum. And here's a picture of the back of the eye. And you can see it has a white tapetum. And that's because the reflective element of the tapetum is actually in these cells called the pigment epithelium of the retina. And the reflective element is made up of lipid droplets. This is an electron micrograph showing lots and lots of tiny little lipid droplets, which gives that an opaque white and reflective appearance. They're the only vertebrate that I know, only tetrapod vertebrate that I know of that has a retinal tapetum. Forget that. Fish sometimes have a tapetum, and when fish have a tapetum, it's usually a retinal tapetum. It's usually contained within the retinal pigment epithelium. And the most common form of tapetum in a fish is a tapetum made up of the crystallized form of the amino acid guanine. You can detect guanine by looking using polarized light when you're looking at the eye. So this is just an example of a retinal tapetum in a fish called an arowana. This is a walleye showing the retinal tapetum in the back of the eye here. This is something called an elephant-nosed fish, which lives in murky water, and there's some other importance of the way this tapetum is derived that I'm not going to get into. And the sturgeon is a primitive bony fish. It actually has a choroidal tapetum. It's in back of the retina, not in the retinal pigment epithelium, but again, it's made up of guanine. Guanine, if you catch a fish, you know that a lot, especially marine fish, have a silvery shine to their skin. And that silvery shine to the skin is also made up of reflective guanine. Okay, so let me just quickly check what time it is. It's been an hour. I made a goal to make this talk an hour. I still have a couple more topics to go. Why don't we maybe take some questions and then people can comment as to whether or not they're in for more or they've had enough comparative anatomy of the eye. How does that sound?
I
David?
had a question about that first. Toward the beginning, there was a picture of different birds with different levels of visual acuity, and human beings were toward the right. The units are CPD. And is that cones per degree? I don't know what that is, but I'm wondering, like,
Nor do I.
I'm wondering, like, in animals, like cats that are adapted more for darkness, if you had to put their vision in, like, is it like 2200 in terms of they could see the same stationary object like at 20 feet and we could see it at 200, or is it
Yeah.
any
I
way
should
to
answer this, and if I'm remembering correctly, cats are really an example, the sort of characteristic of mammals in general. Their visual acuity on that scale would be about 20/60.
and they have better night vision, which is consistent with the
They have better
the
night
trend.
vision, but it's still not understood exactly what that means. It's probably not visual acuity. It's probably, I keep harking back to motion detection, but there are other aspects of vision that might be more important for dim light vision.
Dick, I have a question. Are there any successful animals that have no eyes, or one, or more than two?
So if I go on to the very end, I'll start talking about animals that have reduced eyes for one reason or another. And there are certainly animals that live in total darkness that selection pressure has caused them to abandon their energy consuming eye tissues altogether and have no eyes. There's even more species that have eyes that are embedded underneath the skin, perhaps maintaining some ability to detect daylight length. And then another animal that has reduced eye function would be an animal that has adapted other mechanisms, echolocation in bats, for instance, electrolocation in some fish that like electric eels and what have you, that in the duckbill platypus, that bill structure is a vibration detector and also an electrode detector. So the duckbill platypus has very reduced vision, reduced eyes, although still functionally sophisticated eyes.
And would you classify some bugs and insects that have these multiple eyes that they can see many, many different views at once, like a bumblebee, for example, or a fly? Are those
Yeah, I think it's a
multiple eyes, or
I
just
think it's a
one-
misconception. I think it's a misconception to think that they have many views of the same object at once. I think it's more likely that the image that they form is like a pixelated image, and that each eye is interpreted as a pixel in the brain. The individual eye structures are referred to as ommatidia,
So
and
it's not like they have a retina with a lens on the front that offers
Well,
multiple views.
they
They
do,
have
but
multiple pixels.
they do, but they don't cast an image from the lens. The lens is a light gathering structure, which sends the light back into the tubular structure of the ommatidia, and then the photoreceptor cells in the ommatidia are not. They don't form a film like the retina. They all analyze the light as the light passes through them. So each ommatidia would evaluate the light that passes through it in much the same way a television screen pixels would represent different aspects of the light that go through
So they
them.
don't really form an image like we would think of an image.
That's a good way of saying it, yes. They form an image in their brain for sure, but that image in the brain is not dependent upon an image cast on the retina. So the animal that has the largest eye relative to body size is the dragonfly. And the animal that has the highest degree of visual acuity relative to body size is the jumping spider. Any other questions?
What animal has the largest eye, just in terms of its size?
Although nobody has a good example of it, it's the giant squid. People
How
only
big are
have,
they?
people only have rotten images, rotten specimens of beached giant squid eyes. Well, they're described in lay literature as being the size of saucer plates, and who knows what that means, or soccer balls.
Hmm.
Who knows really what that means? There's an image on the internet of a very famous comparative ocular anatomist holding the lenses, only the lenses from a giant squid in his hand, and you might entertain yourself by searching for that image.
Good. Well, thank you, Professor. Any other questions?
I'm happy to stop here. I will save some material. Maybe in another year or so I can do another talk carrying on from here, or maybe I'll concentrate on the duck-billed platypus because as an evolutionary specimen, it's extremely interesting. I don't know, a number of different things that might be interesting, that people might find interesting. Thank you for being a good audience. Great questions. I'm gonna
Yeah,
stop.
thank you very much. On an organizational level, oops, everybody's gone here. I was just going to say, thanks for putting this together in this format. And maybe that's a good way going forward. So at least for the moment, well, we have no other choice.
Okay. I miss Chinese food.
Yeah.
Yeah.
Thanks, everyone.
Good. Good night, and we'll
Thank you.
I'd just like to say we had, at the peak, we had 17 people. I think I counted 3, count more than, so 15 individuals have attended, and now we're down to 11. So I think we had a good group here tonight, and I think the timing was about right. If we go more than an hour, some people have dropped out.
Yeah.